Nice to see you and to have you here with me for our one of our webinars on today's topic of environmental monitoring and bioburden control in light of Annex 1 that you can all already see. My name is Johannes Oberdörfer and I am your host for the next 30 to 40 minutes on this topic. So happy that you joined and let's get started. The topic of environment monitoring and bioburden control is something that is driving me throughout most of my career. So I joined Rapid Microbiosystems as a field application scientist 2 years ago, and before that time I was already part of a project team that was doing the gap assessment for the Annex 1 that was at this time point in the commenting phase, so that we had already a draft version that we could assess. And aside of this, I was responsible for rapid and automated technologies. So I was working with the Growth Direct system that is distributed by my current employer, Rapid Microbiosystems, which is an automated incubation and enumeration system. If you want to know more, you can find on the website all the informations and details about the system and Rapid Microbio. So now for this topic of EM and bioburden control in light of Annex 1. So just a short disclaimer that this information or these things that you see is like my own opinion. It's not something that is representing Rapid Microbiosystems, my employee, as a whole. This is really something that's from my perspective, and that's also important. So please don't listen, look at it, and then go out into your facility and shout out, "Oh yeah, this and that needs to be done." Double check your information and then get to action if needed. On the agenda for today, so first of all, we would like to understand a bit more on what are the environmental monitoring and bioburden control, and especially also what they are not. So to have a clear explanation or thought on what they are and what they are not. Then we want to definitely look into what does the new EU GMP Annex 1 say. So what are the requirements, recommendation changes? And then at the end, we really want to briefly look into, so what is now my action item? What should I do? Or hopefully, what have I already done? And if not, then what should I do now at least? So what are environmental monitoring and bioburden control? So definitely they are not holistic. And this is something that I found out with many different companies, that there are always the thought that if I have a EM regime and if I have something in place, then I am in control of everything. Yeah, in best cases, this might be true, but generally it's not. It's not a 100% insurance for sterility, especially it depends on how it was designed and how well is it executed, because they are only effective if we consider that they are adequately designed and that people that are adequately trained follow it. So you see already at the start, we see major topics that can conclude that this is not the perfect thing at all because humans are involved. They also are not, definitely shouldn't be a replacement for bad practices and GMP incompliances. Just having them doesn't replace or doesn't substitute for bad practices and incompliances. They are definitely not universal, so there is no one-fits-all solution. There are different facilities, areas, different countries, different situations, even in one facility or in one site, that lead to the point that this is not a universal thing. And if you might think of your own SOPs and like local or global SOPs, there will be so many exceptions and additions and like optionals. That this is definitely not a universal topic. They are sadly not fully explained and also not step by step explained in regulations and in regulatory documents. So I know that we are all looking forward to have something that we can really follow like a recipe, and going through like step by step, first add the flour, then the milk, then the eggs. Yeah, but it's not the case. It's really something that we have to evaluate, and that we have to see how these different parts that the regulations gave us, and laws gave us, how can we follow them, and how can we best suit them and apply them in our settings. And the other one that is important is that environmental monitoring is definitely not environmental control. I think the microbiologists might know, but definitely is something that came across quite often, that these terms are exchanged, and mixed up quite frequently. Then, what they need to be. Definitely, they need to be a part of the contamination control strategy, so the CCS. And I think some of you, like, this is maybe a buzzword or trigger word. When they hear CCS, contamination control strategy, everyone's like, oh, shrugging together and getting a chill, because this is like the holistic strategy of the contamination control, where we spend an awful lot of time to design and to implement it and to fulfill it. And still, as we all know and as we all deal with it, there's also no clear, perfect guidance. And on all conferences that I've attended where this topic came up, there's always the question for, please give us guidance, give us advice, give us examples. But yeah, this is very difficult. So contamination control strategy is something that needs to be as well evaluated and set up specific for the circumstances. EM and bioburden monitoring need to be risk-based. So yeah, we need to assess, where do we have the risks, where do we see potentials. We will dive into this later on. They should and need to be adapted to local settings. So again, not taking a solution from one side and just adapt it and lean back and say, yep, fine, checkbox clicked and checked, everything's done. Definitely not. You need to adapt them. They need to be well documented. Well, should be clear and self-explaining, because in GMP routine and in the GMP production environment, we document well and we go for the good manufacturing practice. So therefore, also these parts of the EM and the bioburden control need to be well documented, and then periodically reviewed. And well, at least from my understanding and from what I can say, is that periodically review doesn't mean that you look at a 4 years interval onto these things, but you look on these intervals in a meaningful and reasonable interval section. So if you review your EM monitoring data on a monthly basis, on a bi-monthly basis, a half-year basis, maybe you have then the conclusions as well, and you should also review your EM and bioburden program in the same periodical interval. But definitely, something that is not longer than 1 year or annually, from my perspective. So what is the purpose of all of these EM and bioburden monitoring program at all? So not only to collect data and to have them. Some data analysts might think, or statisticians might think like, hey, this is good to have a lot of data. Yeah, correct. But not only the data collection, but also to trend, to monitor, and to give feedback, to evaluate it. It's important. So with great data comes great responsibility, and you have to evaluate it, because what do you do otherwise with the data? You should show and maintain different cleanroom environments with the EM and bioburden program, that you are always knowing if you are in control or not. With the data and with the programs, you ensure the release of sterile and clean batches. That's a main purpose for a pharmaceutical manufacturer. Enable appropriate investigation and processes and actions, so as at least also in a timely manner. So it should be fast, should give you feedback from areas and from sampling points that are critical. We ensure with these that our cleaning and decontamination processes are accurate and are effective. If not, then we will also get the feedback that we have out of control in our environments and in our clean rooms, and then we need to adopt. They provide information about the environmental controls, so the different entries and different medias that we have to control. And then we also understand with these programs are the performance of people and equipment. So otherwise it would be a black box, or we would just always look after something or hunt down something that we don't know where the contamination came from. And we are constantly trying to find it. But at the end, the EM and bioburden programs ensure that we know holistically what happens in our facilities. It sounds like they are, and we had it at the beginning, they are definitely not holistic. So we have limitations. And the limitations come either from different, they come from different sources. So first of all, we have recovery and collection efficacy. So if we think of, for example, swab testing, so we have a swab testing, and then what is the method of recovering what we just swabbed? So do we take the swab and then just streak onto our media plate, or do we filter and reconstitute the swab? There are not definitely nice publications on these efficacies, and there are clear feedback that reconstitution and filtration is 60 to 80% more effective, or giving you more recovery than just streaking onto another plate. So that this shows that our recovery and collection efficacy and the testing efficacy is affecting our results. Same for the culture media, that different culture media, different incubation temperatures might conclude to a different microorganism spectra. At the end, the monitoring program is limited, and we all know that it is not showing everything and gives us a complete picture. And there are also publications available that say like between 2 to 6% of the actual contaminations are just covered by our monitoring programs. And if you think of the sampling sites that we chose, this is also just a reflection of, or after risk assessment, a best practice approach to find the most possible contaminations or sources of contaminations and spots that are highly frequent. The monitoring program is also limited by the time of sampling. So whenever you sample right after cleaning, you might find not that many microbes or contaminations at all. If you would sample before the cleaning process, this would at least show the worst case approach. So we can and we are affecting the outcome of our results by the time of sampling. Well, and then the limitation is also timely feedback. So with automation and with rapid methods that give you feedback within one day, this is something where we can ensure a timely response to what happens and what we find. And otherwise, we just have to wait until 5 to 7 days until we get the feedback on, this spot where 20 people daily walk through is contaminated with mold. Yeah, okay, good to know now, but too late. So looking into some solutions for this is critical. Yeah, and the outcome is monitoring is only a snapshot. So we just take samples weekly, bi-weekly, maybe daily, but it's still only a snapshot. The effectiveness of all of this comes with the trends over time and this examination over time. If we have an endpoint measurement, this gives us a good snapshot, but at the end, we only know also if we conclude all over time what happens or what happened in our clean rooms and in our environments. Yeah, what is a good monitoring program? A good monitoring program shows that the monitored area is under control. So we should show what is most likely to be a contaminant, where we would find it. So this is the job of a good monitoring program. It gives us a immediate feedback. Here it said timely, but it would be an immediate feedback as soon as we have an excursion or a trend, or also see a contamination that is above a certain level. It supports the timely and comprehensive investigation and action. So it should be set up to give us these feedbacks in a timely manner and in a way that we don't need to investigate and dive into these topics further into the data. So the monitoring program should give us all the information that we need in one go. It also supports the continuous improvement of the cleanliness and reduction of limit excursions. So the essence is that a good monitoring program, I would also say a normal or standard monitoring program should not ensure a steady state, but should show and give the opportunity to improve and reduce contaminations and limit excursions over time. And this is what is also expected from the regulatories and from the Annex 1 to the manufacturers. There's no expectation that we maintain the steady state and that the limits that they give should not exceed it. That's clear. But the expectation is that we strive to be better and continuously improve or have less contaminations, to get more clean areas and lower warning limits, and adopt the warning and action limits to these trends to further reduce the risk of any contamination and therefore increasing quality of the product and the patient health and the patient security. There are multiple regulatory guidances. So we do have, we can go through all of these, but definitely it shows that it is not a harmonized or one-fits-all of a holistic approach. There are so many guidance documents that support or that give guidance, but not that they state, okay, if you go with this and that, then you're all done and everything's fine and everyone will approve. So at the end, you need to adopt to what is out there, to investigate, experience, and develop from these different documents what is the best strategy and program for you. There's no holistic or universal solution. It's just for your situation that needs to be adopted. As already said, some requirements are clear. So EM programs should be documented within a SOP. Because it's in a GMP environment, we need to follow clear instructions. And the limited guidance is on frequency and contents of the programs. So we have some feedback on the limits, and we have on the alert and action limits, but there's no clear guidance on like weekly, daily, every hour, not holistically. So at the end, we have to facilitate and accommodate the facility type, the facility age, the design of the facility, operations that we are carrying out, different areas that are classified. If we receive some regulatory feedback, and also our statistical power and assessments, the experience, the monitoring period, all of these things lead to the point that we can then determine the content and frequency of an EM program. So you might think like, okay, this is now where the cat bites itself into its tail. So like, where do we start, hen or egg? What was first? So do I first have to monitor data and gather them to develop an environment monitoring program? The answer is not clear, but there is best practice approach where we know that like we assess and we set up a EM program inside of a contamination control strategy. And then again, it's a periodically review. And if we don't have enough data to provide what we should do, the periodical review should be very tight and in a short frequency. And as we advance, it might get longer, but then facility age comes into play, or maybe different products, also equipment that we are using, monitoring equipment that we are using. Maybe there's the possibility to adopt, and this is where these programs and the contamination control strategy live from, that we adopt and that we review, and not only looking into a set stage and we never change. I know most of us would like that we establish something and then we stay with it, but it's not the way it is. The world keeps turning, and things develop in a pace that at sometimes we are struggling to follow. But yeah, that's how the world goes on, and we need to adopt to it. At least the EM program, what should it contain? And there are also some guidance on this one. So the monitoring methods for sure, the media, the incubation method that we are using, the sample locations with a map should be included, the monitoring duration, so how long do I monitor, what do I do, when do I do it, and then RACI metrics, so responsibilities, accountabilities, yeah. So whose role is what? And these RACI metrics is also emphasized in the Annex 1 with clear divisions and clear responsibilities within quality assurance management, quality control, which give a clear direction on what needs to be done and who's responsible for these things. It should contain alert and action levels. Well, without them, it would be a bit meaningless if you could do anything. So what are we doing if we exceed an alert level? What are we doing if we have an action level excursion? Well, and then also the investigation process at the action level excursion should be contained. The other things that are not highlighted here, they are not to be neglected, but at least like these bold marked things are very important. Yeah, as mentioned. So then sample locations, how to select them. And this is in all of the conferences, always a hot topic, like how do we best select the sampling locations? So there are guidance documents like the ICH Q9 or the ISO 14644 and other documents, and also from like external sources, some advisors that help with these. We should use a risk assessment. We should evaluate what is the right sample location. And there are different methods that can be used, like the FMEA analysis or the HACCP. But there's clearly also the feedback on this risk assessment that you should be clear on your calculations, and you shouldn't have an outcome before you conducted the risk assessment. So thinking of a clean room or maybe a locker, if we think of, okay, where the people entered, this is a risk, this is the highest critical spot, then already you failed with your risk assessment because you concluded before you even did the assessment. So it's hard not to get into subjectivity, but you need to maintain objectivity with these risk assessments. Otherwise, this is not representative and it will lead you to a wrong conclusion. And this is the second important part, that we need to have a conclusion and not only where we focus on. And okay, now we defined the risk, but what do we do with the risk and with these outcomes? Generally, there's no right or wrong. At the end, you have to justify and to show that your selection was reasonable and gives you also the required information with the part of the risk assessment that I explained. So don't be driven by your subjectivity, maintain objectivity, and also get into the mode of how can I substitute the risk, how can I, or what action would I take if this risk would occur. So here, just an example of where you would put down the sample locations. You would have a map of your area, of your manufacturing, of your material workflow, and then you would assess, and for each assessed location, you could get into the FMEA analysis, HACCP analysis, and see where you get out of it. And you can and then evaluate what is critical or not, and what needs to be done, and what monitoring you need to be done, and so on. This baseline would give us what was given to us. So now it's about the new, not so new anymore, since 2 years now in place, the GMP Annex 1. So what does it say? In a nutshell, there are more quality risk management processes. So we should do this in a proper way. Auditors already gave the feedback that We shouldn't just do risk assessments for the sake of risk assessing and meeting people in a project team. We should do this as required and as meaningful, but sometimes we just also need to implement an additional sample or an additional control instead of a risk assessment why this is not necessary. So people tend to get through with these things with the risk assessment quite heavily. Less human intervention in the manufacturing line. This is what was introduced. We have faster detection of contaminations in EM and products, and a higher QC testing volumes for the microbiological lab. So in a nutshell, so this is really like condensed if you would ask what changed in the Annex 1. And depending on your field of expertise, you would disagree and say like, oh yeah, but you know, there is the change with isolators, there's a change with the PubSit and so on. Yes, all true. Yeah, but this is from my perspective and from the topic that I deal with. This has been stressed, I think, quite often and showed to you, and you all know. But since the Annex 1 in 2018 version, we've gone through a lot of iterations and a long way until we have now a document with 57 pages, more than 6,000 comments, and you see it like a total change of 1,362. So there has been a lot of change, and there has been a lot of additional information in the new Annex 1 with an increase of 41 pages to 57 pages now from 16. So with the guidance also on specific topics, now it's nicely structured into these 10 topics, which are scope, principle, PQS, the premises, equipment, utilities, personal, production, specific technologies, environmental and process monitoring, and quality control. And not to forget the glossar at the end. So what are the content of these things? And I will just really dip into this one, not going into details, as you can read this through afterwards as well. And there are more detailed presentations and webinars available that will go into the specific topics that you would need to know. One part of it is the clean room and clean air, that we have these three topics of classification, qualification, and monitoring. So it's clearly what is classified as clean air, how do we qualify these things, and the monitoring of the clean room and clean air, and also then the routinely monitoring of it. So there has been some changes to the particles and how we monitor these things. More details later on. The principle emphasizes now on the use of rapid and alternative methods, which is like with my rapid microbiologist heart is a great change. That overall, like in 10 to 11 positions in the Annex 1, it's mentioning automation and rapid methods. So emphasis on the use of these is important and, well, state of the art to use it. I don't think that it is state of the art to use a gel clot instead of LAL test. Yeah, and then we can discuss if a LAL test is state of the art if we have a recombinant factor technology. So these things, just as an example to give to you. Then, well, we have also that in QRM that we should include appropriate design of facility, equipment, and processes. And if you might have wondered, so I have here a commented version and snippets of a commented version of the Annex 1, where we have in blue completely new additions and in orange something that was rephrased or changed. Yeah, well, and then in principle, there was also in the 2008 version, the point of the CCS should be actively updated and should drive continuous improvement. That's for sure. But now we have a real, like, more sentences, more content to it, where it emphasizes again on you should continuously improve and not just stay at the level that you are. And from the experience, it's more we try to maintain what we have, but we do fail to improve and to get better in this monitoring. The contamination control strategy is a big portion of the Annex 1, where we now have also clear definition on what is the content of the CCS. And also in the assessment of alternative methods, that you should assess why you are also not using these technologies. So the CCS contains many different parts, and it's we could talk 1 or 2 hours about the CCS on its own. But yeah, you see here in these bubbles what the Annex 1 defines as part of the CCS, and that is mandatory to be described in the contamination control strategy. And then also well-known picture with these house of where we have the contamination control strategy, holistically looking at it, where we have the foundation with the contamination risk assessment and the quality culture, which we build upon all the following parts, where nearly at the top we have the monitoring of these controls. Then with the premises, we have the smoke study requirement, which are heavily emphasized on, and a lot of companies had to redo or to do these smoke studies in order to be compliant with the Annex 1. Then cleaning programs need to be effective to remove disinfectant residues, which is also not always as common as we might think of in companies. Well, and then studies should include representative surface material, that you have these materials tested, that you have in your facilities. Then in the premises, we have a table. So in the section 4, there's a table, the table 2, with maximum permitted microbial contamination level. Just be aware that this is like a table that represents under qualification. So during the qualification phase, this is not the actual monitoring table, because that was something that was brought up several times with customers. The real monitoring table and the explanation to this table is in the section 9 in the environmental monitoring, the table 6 under point 9.31. Yeah, then barrier technology. So I don't go into the details, but you see that this was completely revamped with all the yellow. So these parts of isolators, wraps, the closed isolators, open isolators, was completely overworked, and there are more better experts on these topics you just have to look for the vendors, and you will find a lot of more information on these parts. Oh, sorry. Yeah, and then also in the closer section, we have a bit more of a detailed explanation about the closed and open isolators of the wraps. And clearly the guidance is towards have the isolator system and not introducing wraps and these technologies to justify why you're using this. So this is a high emphasis on within the Annex 1. Yeah, then clean room and clean air equipment qualification. I already mentioned this at the beginning, is mentioned here with the different explanations. So just to note that on the right side, we have the snippet of the 2008 version, and on the left side, we have the 2022 version of the Annex 1. Then also there's the total particle concentration mentioned, and also the potential loss for particles within sampling systems. So yeah, as mentioned, I'm definitely not the expert on this one, but yeah, the vendors for specifically the airflow controlling, the active air monitoring, particle monitoring, they know how to best explain it and to support you. So there's definitely your go-to address. Well, and then also on the section 5, the equipment, where we have the points. Grade A zone should be monitored continuously. Sure, sampling methods and equipment should be fully understood. This is nothing new. There's just more emphasis on these parts, especially on continuous monitoring systems with the manufacturer's recommendations on specifics for diameter and the bend radii. Utilities. Results need to be verified before batch release. I think it should be clear and nothing that needs to be told. Alert levels should be based on initial qualification and periodically reassessed. and review of ongoing monitoring sampling programs reflected in the CCS. All the parts that we were talking about at the beginning here written down in the Annex 1. Then your beloved socks from outside are no longer allowed in grades B and C gowning rooms, changing rooms. So yeah, I'm very sorry for these fellows that they need to stay outside of this area now. And regular disinfection of gloves during operation. Laundry facilities should not introduce additional risk. And maybe this point is important to look into all external outsourced parts. Do we have control over them? Do we know what they are doing? So there are also feedback from FDA and from other regulations on the evaluation and the knowledge from us as outsourcing party to the outsourced party. So you don't hand over the responsibility to the outsourcing to the CRO, to the CMDO. It still remains with you. If you thought that everything you can just point down to them, that's definitely wrong, and you should rethink the strategy of outsourcing and your knowledge of the quality system of the outsourced company. Well, yeah, in the Section 8 production-specific technologies, there's a part of additional bioburden product testings where it makes sense and where it's designed so that we can introduce and can look into other steps throughout the bioburden testing. And then the blow-fill seal, alignment with the site CCS, plus additional aspects that are listed here. You see blue, this means like completely new and redone. Have a look into this one. A heavily discussed part is the PubSit, so the pre-use post-sterilization integrity test. And in every conference, it's a hot topic, and there's also no clear like guidance or understanding, not from the industry and not from the regulations. So there's still a lot of room for discussions, like what are now the requirements? What do we need to do within the validation, and how do we implement and use existing facilities? So yeah, heavily discussed at the moment still. Then we have here for the environmental and process monitoring some summary. So this is the largest section that addresses also humans in class AB. And we all know this is the main source of contaminations. There's also reclassification of staff activities outside of the clean rooms. Then it is emphasized to use more of barriers and isolator systems, robot handling, so getting the people out of there. It urged the improvement and training of the personnel, specifies microbial testing of hands, arms, chest, foreheads, and limits maximum gown time. So this is what it emphasized. More change that also equals more testing. At the beginning, we have already like establishing an environmental monitoring program as one part of it. The trending and continuous monitoring, that this is expected, so that we are not doing it just case by case, but that there is a trending and continuous monitoring in place. We have the risk assessment where we determine critical monitoring locations, air visualization studies, like the smoke studies mentioned, and that we regularly and periodically review them. And here there was just an example of what helps us, and these are air monitoring softwares and programs where we know which spot, a picture of the spot, the limits are there, how often do we monitor, and that we see already with one click, oh yeah, we are well within the limits or we are out of the limits. And this is also supported, this software are supported with automation, with automated enumeration and incubations, where it automatically feeds and gets fed by the systems, where we don't have paper, and you all know the positivities of being paperless. And then we have also more stringent action limits that are expected and should be established based on the results during qualification and the periodical review, which I mentioned at the beginning. One part is this static, dynamic, and continuous monitoring. So viable particles, one part of it that needs to be monitored. And you see here in table 1 the maximum permitted microbial contamination levels, which changed in the grade A from below 1 to being clear that no growth is expected. Continuous monitoring is expected, as mentioned. Glove monitoring after each critical intervention. You see the blue part, which is new in the Annex 1. Manual procedures and where persons are involved in grade AB microbial monitoring is necessary, and the quality unit has the oversight. For mold trending, there's like a bit of a different interpretation to it, where you can see in the 9.31 that the end that following the isolation of organisms that may indicate a loss of control, deterioration, cleanliness, or that may be difficult to control, such as spore-forming microorganisms and molds, and at a sufficient frequency to maintain a current understanding of the typical flora of these areas. This part is combined with identification of microorganisms that we detect not only in grade A, B, but in grade C, D, and not only where action limits or alert levels are exceeded. So an important part is that these things are more and not only at the action level. So whenever a mold is detected, you should do and go into the action mode and not just wait for like, oh yeah, well, our trending data says like 4 is okay, so we have an action level. That doesn't make sense. One mold is sufficient to go into action, and you should go into action because you know yourself what this does mean if you have a single mold contamination and you're spreading this out into your whole facility. So don't underestimate the value of this last sentence of in the 931. Another one that is important is the table 6 that I mentioned for the viable particle contaminations. If we look into this one, there is a note section that emphasizes again, and here you can read it, it should be noted that types of monitoring methods listed in the table above are examples, and other methods can be used provided they meet the intent of providing information across the whole of the critical process. This is again a point where automated alternative methods and rapid methods are introduced and the doors opened for you to step through and use it. And then support and assistance to parametric release, which is important in the section 10. Parametric release is powerful, so you should consider and use it where possible, especially with rapid methods that give you feedback in a short amount of time throughout your in-process control process, throughout the manufacturing process. Clarification on growth promotion tests for all product-related testings is here, and we have EM data review as part of product batch release. There are also additional parts that we have the APS, the aseptic process simulation, in this periodic verification that is included. Training status and regulatory confirmation of employees is throughout the whole document very important, and that alternative release test methods for products without possibility for a sterility test, the short shelf life. But also the Annex 4 describes these things very nice, or at least gives guidance. Now then, a summary of changes. Quantity and duration of quality control tests required of laboratories might have increased already, and if the interpretation was done properly, then it should have been increased. EM programs must apply to the QRM standards to facility design, equipment, process, procedures, monitoring systems. Continuous air monitoring is expected in grade A zones and recommended in grade B zones via settle or passive or active air and other methods. The state of control must be maintained throughout processing. All colonies from grade AB must be identified. And again, not only AB, but if you find molds and if you find microbes that indicate that you are not in control anymore, then this also should be done in C&D and periodically anyway. So this is really important. And VFI systems may require continuous monitoring of TOC and conductivity indicators rather than these discrete sampling. So the summary again, more quality risk management, less human interventions, faster detection of contaminants in environment and product, and higher QC testing volumes for microbial labs. At the end, what do you need and what do you need to change, adapt, or do? Well, sadly, if you expected something that says like, you have to do X, Y, and Z, that's not going to come. So for the Annex 1, The implementation deadline was already last year in August, with the exception of the loading testing. This was until this year, 2024, August. So now you need to comply since last year already. So I hope that everyone did already perform a gap and risk assessment comparing current processes and contamination control strategy implementations with the new Annex 1 requirements. So if you did not do, you should do it, like start immediately. And then maybe I have to get back to the point at the beginning where I said you shouldn't run out screaming that we should do X, Y, and Z. If you didn't perform a gap assessment and a risk assessment, then this is your moment to run out and scream and shout. Review, draft, implement the CCS, and consider the use of appropriate barrier systems and processes. So yeah, really rethink your assessments, rethink your EM program, start with your contamination control strategy, look through the Annex 1, look into what does it emphasize on, take the big chunks, split them apart, go into these details. The expectation from regulatory, and this was this year on many different conferences, was clear. Now today you cannot hide behind something like, oh yeah, well, we didn't know and we were just waiting for some feedback. So if you didn't perform any assessment and giving some guidance to them what you are doing and getting also into action mode, if you identified gaps, then you will have a tough time. Starting with this, if you did not do already, is a good point. It's recommended. Yeah, at the end, the only point last for me to make is really use and get into, there's so many expertise out there from vendors, from different parts of this. And I never met someone that was not willing to support and help with the contamination control strategy, with the assessment of the Annex 1. So feel free to reach out to them, use the knowledge, reach out to other colleagues, to the network in the pharma industry to assess and to get best practice examples. And at the end, define your own strategies and assessments. I have to thank you for your patience and for your time listening.